Hello, everybody. With great pleasure, I would like to talk to you about some natural resources, pharmacological properties, and the active constituents in dietary intake applications. So the aim of this lecture is to show you some examples about what we are eating and what we are drinking and how much it benefits our body and how we can overcome the big problem of COVID-19. So this is some examples which we are going to discuss in our lectures. Let us start with the bee products. Bee products contains not only honey, but also royal jelly, propolis, bee pollen, bee bread, bee wax, bee venom. So this is just some literature data about the problem. What is the problem and how we can overcome the problem? According to World Health Organization, 2 billion people will attain the age of 60 years or more by 2050. But what is the problem? The problem is brain aging and neurodegenerative diseases like age brain, Alzheimer's, etc. And how we can avoid this problem. Of course, these are the main problems, but also there is another problems regarding respiratory, kidney, cancer, eye diseases, inflammation, etc. Let us take some examples with literature data. For example, if we start about learning and memory, if we really eat honey, this will be a very good treatment, improved both short-term and long-term memory. And these improvements were attributed to neuronal proliferation, like these examples of some published paper in very good impact journals about honey effect regarding such diseases. And this effect of long-term honey, sucrose, or sugar-free diets on memory effect. What about Alzheimer's diseases? If we take, for example, the ingestion of propolis more than 12 months protected elderly people living at high altitude from congenital declines. Also, oral administration of royal jelly alone or combined with ginkgo biloba extract, this is a trade name, Mumu, is beneficial in treating the congenital declines. Next, about Parkinson's diseases, PD. If we take like bee venom, it reported to normalize neuroinflammatory apoptotic markers and restore brain neurochemistry after routine injury. And this some papers to show the effect of the bee venom. If we take multiple sclerosis, MS, then honeybee venom reported to have a positive impact on multiple sclerosis in humans. And bee sting therapy is increasingly being used to treat patients with MS. Additionally, in clinical trial, people with multiple sclerosis progressed from needing a considerable amount of self-care to a minimum amount after treatment with the venom. This published in Toxins and other papers as well. What about how to train your brain to avoid anxiety? This is again, consumption of honey was reported to reduce it in rats upon oral administration as shown in vivo studies. An enzyme treatment of royal jelly also supplement improved ancestry sclerosis in Japanese women. Propolis essential oil was reported also to significantly reverse the effect. And this is a published paper to show about it. What about spinal cord injury? So again, royal jelly can diminish the secondary neuronal damage after experimental spinal cord injury in rabbits. So the royal jelly was neuroprotective activity against paralysis in male rabbits. After 4 hours, it was a treatment of a spinal cord injury. Depression. This is serious problem now. And it shows that royal jelly was shown to affect against depression, and it associated with menopause in rats as per the estrogenic effect of royal jelly. So also they shown the activity related to long-chain unsaturated fatty acids, which has unique to royal jelly, was also reported to have antidepressant-like action. What about the bioactive compounds from bee products that has shown a neuroprotective agent? Like, for example, cinnamic acid derivative, long-chain unsaturated fatty acid, flavonoids, paracumaric acid, cinnamic acid derivatives, and flavonoids. We recently published a very good paper which shows that such effect about honeybee product, an updated review of neurological action, and it has been published in Trends in Food Science and Technology with several countries. We also studies about Mellitin, which is the main active pharmacological active constituents of bee venom. So Mellitin, as you can see, it has 26 amino acid peptide. It is the main active pharmacological constituents of bee venom. Why? Because this peptide comprises about approximately 50% of the dry weight of bee venom. So it is water-soluble peptide, and the molecular weight is 2,840. And this is the chemical formula of the structure. What about the effect of this Mellitin? It has shown several important effects as antiviral, antibacterial, antifungal, antiparasitic, and anti-tumor. It also showed oxidative stress. It is important, and we will show some of our work on Mellitin, like apoptotic activity of Mellitin. If you see the apoptotic activity of Mellitin and the effect of Mellitin on B-cell lymphoma too, then you can see that this is a normal of the cells, and this is BQ. This is BQ plus Mellitin at low concentration of 0.1 milligram per kg. And when we increased the concentration to 0.5, it shows such effect. Again here, with normal in the survival, and this is BQ plus Mellitin at 0.1, and BQ plus Mellitin at 0.5. The antioxidant activities of Mellitin also we studied it, and as you can see, this is a normal, and BQ plus Mellitin at 0.1, low concentration, and at high concentration, not much effect. Similarly, the PQ and at 0.1, while this at 0.5 effect. Therefore, there is antiviral properties of bee venom and its components, which is mainly Mellitin, and it also contains phospholipase A2 as a second polypeptide. I will take you to another story about Roselle. This shows this paper, it shows a comparative analysis of high bases Sadifira, Roselle, hot and cold extracts in respect to their potential alpha glucosidase inhibition, which is published recently in Food Chemistry. So, if you see the pharmacological properties of Roselle, it is anti-diabetic, hepatoprotective, anti-hypertensive. It's also antioxidant. But then what is the question? The question is how to drink it, hot or cold, right? Which is better also? Is it the sample from Aswan or from Sudan? In order to prove this, and this is how we as a scientist should interact with the society, and this to see what is the chemical constituents and what is the biological activity. So we took samples and by decoction and also by infusion, and they studied also about the sample when we use it as maceration. And how we study this? By using spectroscopy, like the metabolomics can be studied by profiling, the chemical profiling by NMR and LCMS analysis. Of course, LCMS analysis is more sensitive than NMR because it can show you all the constituents. Then we make combining of all the data derived simultaneously from both platforms, which might provide better insights on secondary plant metabolomics. To study this shows the proton NMR analysis of the different samples. And as you can see here, the aliphatic region and the sugar region, and then the double bond. So this shows the aliphatic acid mainly. And if you go here, it will show the aromatic phenolic compounds. And here it shows you the signals of furfural. So the proton NMR analysis of the extracts, it shows signals to show you the different classes of the compounds, but you cannot prove the single compounds from NMR. But with the compiling of mass spectrometry with a molecular weight and fragmentation pattern, then we could identify different compounds from Aswan samples and from Sudan with different kind of extractions. So this is, we call it chemical profiling. It means that we can identify the compounds in different geographical regions with different kind of extractions. And this study shows the alpha glucosidase inhibition assay of Roselle tea preparations. So with red color, it is Aswan samples, Sudan sample with green color. So, and this shows the IC50 by using different methods of extraction, decoction, infusion, and maceration. So the IC50 values of alpha glucosidase enzyme inhibition assay of the tea preparations expressed an average microgram per milli, and it is calculated of 3 independent assays using a carambose as a positive control. So what is the conclusion of that? Better to use of Aswan sample as a source of Roselle owing to its higher contents of sugar and anthocyanidine levels in comparison to Sudan sample. So the chemical profiling in compilation with biological activity, it shows you such results. And the cold maceration proves to better preserve the anti-hyperglycemic properties of Roselle, while infusion results in extracts more enriched in hydroxycitric acid of potential weight loss effect. So it means that we could answer the scientific question which has been raised in the beginning of the paper. Another example, onion and garlic. This is important to ask ourselves such a question. Is it really healthy to eat such important antibiotics and antiviral like fresh or with treatment? Right? To answer this question, we made such phytochemical profiling. So again, to solve the problem and to interact with the society, we as a scientist should use spectroscopy for chemical profiling and also biological activity to approve what is the difference of antimicrobial activity if we use such onion and garlic as a fresh compared to if we use it in dry, like such tablets. That's why we say phytochemical profilings and antimicrobial activities of allium subjected to different drying methods, comparative MS-based metabolomics. The question, what do you expect? Better to use it like that or to use it in tablets, right? So this is natural result, completely naturally, and this is not completely naturally because you composed it to drying methods, right? Let us see what happened of this exciting question. First of all, if you take onion and garlic, it's very important, not only as anti-cancer and hypertension, obesity, but also in heart diseases, in treatments of diabetes too, right? In order to do identification, we used SPME, and this is to determine the volatile compounds. And when we say volatile compound, it means that we must use GC-MS analysis. Then we could identify 30 sulfur compounds. So this is a sulfur compounds in both, which showed such effects which we discussed. And this is example of natural product classes reported and detected in the genus by LC-MS analysis. And if you look to the compounds, it contains, of course, a sulfur compound, aromatic compounds, flavonoids, etc. Now, it is exciting to know the question, to answer the question. So this is the fresh material, and this is the tablets. When you analyze such fresh materials, it gives the expected volatile compounding containing the sulfur compound, and it means that it has antimicrobial activities, right? Of course, for volatile compound, we use GC-MS analysis. But for non-volatile compound, then we must use LC-MS analysis. Then identified the non-volatile compound like diterpenoids, flavonoids, etc. What about for the tablets after drying? Oh, now we could see the answer. That it shows different type of compound like diethylphthalic acid. It means we don't get the other compound. Accordingly, no antimicrobial activity. So which is better to use, the tablets or the fresh? Of course, the fresh, because it gives you the volatile compounds and with the activities. Now we go to another example about sugarcane. if we would like to make chemical profiling to study the metabolomics, then we should use spectroscopic analysis and then the biological activity of the sugarcane juice and its product molasses as well. And this paper published in Molecules. So this is a sugarcane and uses of the sugarcane and with as well as activity. What about to study the sugarcane? We have two types, the sugarcane and the molasses. Of course, to do the metabolomic profiling, so these are the samples we studied, these two samples, and this is the metabolomic profiling. It means that we will study using NMR, GCMS for volatile compound, UPLC-MS for the secondary metabolomics, which is non-volatile compound. What about the biological activity? Total phenolic content, total flavonoid content, the antioxidant activity, and enzymatic activity. So this is a sugarcane and the molasses, and this is schematic diagram to show you how we can study GCMS analysis for primary metabolomics and for secondary metabolomics, UPLC-MS, as well as the NMR. And this is the biological activity for the study. Now, if you take the NMR in this region, it contains the aliphatic region with long chain fatty acids. And here is the sugar area, right? With the anomeric proton and aromatic region. Let us see the different identified compounds: sugars, and then amino acids, flavonoids, long chain unsaturated fatty acid, and phenolic acids. Now, let us take another example about what we eat and what we drink. All of us are afraid from the second wave of COVID-19. Then how we can protect ourselves as much as possible? Then we must focus on immunomodulatory. And it means that our body is what we eat and what we drink. And remember that eating healthy food, sport, non-smoking, avoid stress, sleeping well, these are main factors to protect ourselves. Of course, in addition to social distance. Let us talk about our area. This is a food which increase immunomodulatory effect. Pomegranate, grape, figs, ginger, olive, garlic. Very important for us. Of course, in addition to bee products as well as other examples. What we have discussed recently, this is the diversity of bioactive compound from natural resources as anti-coronaviral activity. So this is different compounds which has been in preclinical or clinical studies from different species, alkaloid, another alkaloid, and the polyphenolic compound. From another resources, another alkaloid, beverine, alkaloid, and alkaloid, different alkaloid compound. So we had been working hard to collect such data for study what has been done as antiviral in preclinical and clinical studies and how this could be related with COVID-19. So this is another example. And from Artemisia, we all know about artemisinin, and even from bacteria, they isolated this compound, and it is in preclinical studies in both. different other plants of the same genus, but different compound is alkaloid, while this is steroidal compound, and the flavonoid compound, and different type of chalcone compound. From other resources of the plant species, then flavonoid, steroidal compound, and from citrus. This is important, a flavonoid compound and another type of compound. Different species, more complicated structures with stereochemistry, and then another species, steroidal compound in clinical studies, another in preclinical, and from this bacteria, it is preclinical compound. So what is important here after we study such species? This kind of food is very important for us. Why? Because it's important for immunity system. So COVID-19 is hazardous for the people who have a weaker immune system. So this is a point than healthy ones. So strong immune system is essential for fighting against COVID-19. Consumption of healthy diets is one way to boost the immune system. There is a wide range of plants that contribute to boosting the immune system, giving their high content of a wide variety of nutrients and biological active compounds. And this is supported with references, of course. Let us take some examples also about food, like broccoli, spinach, and the sweet potatoes. This type of food, they are abundant with vitamin E, which include retinol and beta-carotene. And these fat-soluble compounds precipitate enhance of immune function. So this is the key point. Additionally, vitamin E derivatives inhibit such COVID, SARS, etc. Garlic is important nutrient with sulfur-containing compounds. It has antivirus, antioxidant, antibiotic, very strong as antibiotic, anti-inflammatory, anti-cancer, and cardiovascular properties. What about citrus? Very important for us for vitamin C. And the vitamin C is water-soluble vitamin that is not stored in the body. So daily doses of about this amount per day is needed. And the vitamin C increases the protection of white blood cells, providing a key role in fighting infections. Vitamins are recommended for the protection against coronavirus as they increase patients' resistance against infection. In conclusion, the biological and conventional food process modifications on food proteins, structure, functionality, and bioactivities. We studied this. And the food proteins, like such proteins, is it healthy or not? It's very healthy and very important. Why? Because it required to maintain various body functions in human health. And this is what we published also in Biotechnology Advances. So this is what I end my lecture about this important paper, which we published recently with different countries, Sweden, Egypt, China, Pakistan, Germany. And the paper, it discussed the screening for natural and derived bioactive compounds in preclinical and clinical studies, one of the front lines of fighting the coronavirus pandemic. It's published recently in Phytomedicine. With this, I would like to end with references about the compounds which I showed as preclinical and clinical studies of antiviral. And I must say that collaboration is very important. It doesn't matter the color, the gender, religion, etc. But we all could collaborate together to be more stronger. And this COVID-19 is an international problem, which means that we all must cooperate together to find a solution. With this, I would like to end by the acknowledgement of Sweden, Japan, and collaborators and cooperations with Netherlands, Germany, Malaysia, and China. You are very welcome to visit Sweden in summer, better than in winter, because in winter it is a lot of snow, and of course, Egypt as well. Thank you very much for your kind attention.